video, we're going to talk about the process known as hemostasis, or blood clotting. It's going to be comprised of three main phases: vascular phase, platelet phase, and a coagulation phase. We're going to start up here at the upper left in panel A. And we've got blood flowing through our vessel here. And we've created a hole in the vessel wall here. We've damaged the vessel wall. And so certainly our blood components are leaking into the interstitial space here, the ISF, interstitial fluid. Now, that would include red blood cells, platelets, white blood cells, which aren't shown here, as well as all of the plasma components. Now, something to point out here are the main components of our vascular wall. That includes smooth muscle cells, at least in arteries and veins, collagen fibers that underlie the endothelium. Those collagen fibers are part of what's called the basement membrane, right? It's the layer of connective tissue to which that lining layer of endothelial cells, shown here, are connected to, right? Now, so we're forming a bruise here, right, over time. But what's going to happen very rapidly in response to this damage is we're going to get what's called a vascular spasm, this vasospasm. This is what we call a myogenic response because it comes from the smooth muscle itself. And damage to smooth muscle leads to a very rapid rise in intracellular calcium that causes it to constrict. And that's going to help, right? We're going to get this constriction, this inward movement of our wall as the vessel wall tightens. And that's going to dramatically increase resistance to blood flow along this particular vessel, diminishing blood loss. That's very helpful right off the bat. But in addition, we're going to start to activate platelets. They're going to release other agents like thromboxane A2 that is also vasoactive, helping to add to this vascular spasm. Similarly, the damaged endothelium is going to release endothelin, also vasoactive, vasoconstrictive, causing to minimize the blood loss. Now, we're rapidly going to transition into phase 2 down here in panel C, the platelet phase, where the platelets are now exposed to the collagen fibers underlying the endothelium. Remember, that endothelium usually covers up that basement membrane. But with the vessel wall damage, the collagen fibers are now exposed and the platelets start to stick to them. And they stick to them through this linkage protein called von Willebrand factor. Right? Normally, those two do not see each other, the platelets and the collagen fibers, but once they do, the von Willebrand factor will help link the two together, initiating formation of this platelet plug. Right? This is not the sturdiest clot. It's called a white clot sometimes, but we have to sort of stabilize it. And so what happens as it develops is we start to trap through receptors on the platelets this circulating plasma protein called fibrinogen. Well, fibrinogen is a precursor to a clotting protein we're going to see in a moment. So we're starting to incorporate these fibrinogen strands within the platelet plug. We're getting the release of our thromboxane. We also get release of ADP, which is stimulatory to platelet activation. And so platelets start to stick, release ADP, causing more platelets to stick, releasing more ADP. Sounds like a positive feedback loop, right? Well, that could be dangerous. We don't want our clot to grow out of control. So once our clot grows into healthy areas, that healthy endothelium releases anticoagulant agents, PGI2, prostacyclin, and NO, nitric oxide. Those tend to diminish or shut off this platelet activation and sticking, helping us to localize the platelet plug to the damaged site. Now, in the last panel down here in E, right, we initiate the last phase is coagulation, right? Some of the factors released during the platelet phase are leading us to this culmination, and we get tissue factor released by that damaged activated endothelium, and through a very complex cascade of enzymes, convert prothrombin to thrombin. This is the essential step. Thrombin is the enzyme that catalyzes the polymerization of our trapped fibrinogen, right? Remember, that's up here. All these fibrinogens trapped, and they're going to polymerize it into fibrin strands. And these end up getting cross-linked, so we form essentially a mesh within the platelet plug, forming now a final clot. Now we've reduced or shut off our blood loss, and we finalize our clot formation by stabilizing it with these fibrin strands.